Okay, let's start talking about the muscular system. This is Chapter 7 in Colville and Bassert's in Comparative Anatomy and Physiology. Our learning objectives today will be to list the three types of muscle and describe the characteristics of each, describe the structure and function of tendons, aponeuroses, and ligaments. Differentiate, differentiate between the prime mover muscle, the antagonist muscle, the synergist, and the fixator muscles. And we have to remember that those will change depending on the movement. We'll list the locations and actions of the muscles of the head and neck, abdomen, thoracic, and pelvic limb, and the muscles of respiration. We'll describe the microscopic anatomy of skeletal, smooth, and cardiac muscles. And we'll list the components of a neuromuscular junction and describe the function of each. We'll also describe the events that occur in muscle cells during muscle contraction and relaxation. And it's pretty cool to see how things happen. It kind of looks like a factory setting within your muscle cells. We'll also differentiate between visceral smooth muscle and multi-unit smooth muscle. All right, so muscle is one of the four basic tissues of the body. It's made up of cells that can shorten or contract. And there are three different types of muscle. And can you tell me what they are? I actually just named them in our learning objectives. There's cardiac muscle, there's skeletal muscle, and there's smooth muscle. Cardiac muscle is branching fibers. Skeletal muscle is one longitudinal fiber. The muscle cell is just one longitudinal cell that join together in fibers and then make up the muscle bodies. And then the smooth muscle cells look like spindles. Cardiac muscle is obviously in the heart, whereas skeletal muscle is in the movers of the skeleton, and then the smooth muscle cells in the organs. Skeletal muscle gross anatomy, the things that you can see. So let's differentiate these or describe them. Tendons are fibrous connective tissue bands. They connect your muscles to your bones. Aponeuroses are sheets of fibrous connective tissue. So in your palm, you actually have a sheet of fibrous connective tissue called the palmar aponeurosis. It comes from the tendon of the palmaris longus, which is a muscle in your forearm, and it attaches from the muscle and then turns into a sheet or an aponeurosis and connects to the bones of your finger. An origin or the origin of a muscle is the more stable of the muscle's attachment sites, and usually that occurs more proximally. The insertion is the site that undergoes most of the movement when the muscle contracts. So sometimes when we describe a muscle, we describe it or we name it based on the origin, where it comes from, and the insertion, where it goes to. Muscle actions. Okay, so muscle actions can change in each muscle depending on what the movement is. First of all, there's a prime mover or the agonist. The agonist is a muscle or muscle group that directly produces a desired movement. When you lift your fist, your agonist is your biceps. It's the one that contracts. The antagonist will be the muscle or muscle group that directly opposes the action of the prime mover. In this case, when you raise your fist, the antagonist is the triceps, or it's relaxed. Now, when you straighten your arm, your triceps contract. So that becomes the agonist, and the biceps become the antagonist. A synergist is a muscle that contracts at the same time as the prime mover, and it assists. It's synergistic. It assists in carrying out its action. So when you lift a glass, you are having a major muscle movement of your biceps brachii, which is the prime mover, and your brachioradialis, which is a smaller muscle on your forearm, is helping that the action of raising the glass, and that becomes the synergist. A fixator is a muscle that stabilizes the joint to allow other movements to take place. So imagine if you were raising the glass and all of a sudden your glass tipped over because the muscle in your shoulder that holds that joint steady as you raise the glass isn't working. So that fixator muscle in your shoulder stabilizes your arm so it comes up in one direction and doesn't tip. So muscle naming conventions, just how do we know how to name these muscles? We can name based on the action. For example, calling a muscle a flexor muscle or an extensor muscle in something that extends. We can name it by the shape. deltoid, for instance, means triangular shaped. We can name it by location. For example, the biceps brachii muscle is located in the brachial region, or the direction of fibers, meaning we're naming a muscle because the fibers are straight. So a rectus means straight. Rectus abdominis means the straight muscle. Transversus abdominis would mean something that goes across. We can also name it by the number of heads or divisions. So when we say sep, that means head. Biceps would mean the biceps brachii has 2 heads. We can also name it based on attachment sites. So if we talk about the origin of the sternocephalicus, we can know that the origin is the sternum and the insertion is the back of the head. Cutaneous muscles, these are the first muscles we come across when we're doing dissection. It's actually very difficult to find because they are really attached to the skin. It's a thin, broad, superficial muscle. It's found in the fascia just beneath the skin with little to no attachment to the bones. The only the only action it has is to twitch the skin. So you can think about that cutaneous muscle, the cutaneous trunci muscle in particular, on the skin of a horse twitching the skin. Head and neck muscles, their function is to control facial expressions, enable mastication, move structures such as eyes and ears, support the head, and allow the head and neck to flex, extend, and move laterally. We have the masseter muscle, which closes the jaw. And in our animals, it can be very big because they use their jaws on pretty heavy material. It looks like a big round muscle here in the cheek. We also have the splenius and trapezius muscles, which are on the back of the neck, and they help to extend the head and neck. We have the brachiocephalicus muscle, which extends the head and neck and also pulls the front leg forward. Brachiocephalicus muscle. And so brachio meaning arm and cephalicus meaning head. We also have the sternocephalicus muscle, which flexes the neck and the head. That goes from the sternum to the back of the head. So the functions of the abdominal muscles, so it was head and neck, we're going to abdominal, are to support the abdominal organs, keep them in place, flex the back, and participate in various functions that involve straining and respiration, breathing. They're arranged in layers, and there are 4 that you need to remember. The external abdominal oblique, it's outside of all of them, and it runs obliquely. The fibers run obliquely. We also have, actually they run this direction. We also have the internal abdominal oblique, which is inside underneath the external abdominal oblique, and the fibers run this direction. We have the rectus abdominis muscle, which runs straight right in the middle. And we have the transversus abdominis muscle, which comes down here and runs straight across. The left and right parts of each of these muscles come together on the ventral midline at the linea alba, which is Latin for white line. And it's this nice white line that gives us an indication of where to cut when we're entering the abdomen for surgery. Thoracic limb muscles, these are superficial muscles of the shoulder region. The latissimus dorsi is this great big trunk muscle here. It flexes the shoulder. The pectoral muscles, there's one superficial and one deep. They adduct the front leg, which means it brings it closer into the body. I'm sorry. Yes, it brings it closer to the body. Abduct means to take away. So the deltoid muscle on the back takes away, abducts and flexes that shoulder joint. We're going to find all of these in dissection as well, so it'll make a lot more sense. The brachial muscles you'll need to remember. The biceps brachia, which flexes the elbow joint. The triceps brachii, which extends the elbow joint. And the carpal and digital muscles. There's the major ones are the extensor carpi radialis muscle, which extends the carpus, and the deep digital flexor muscle, which flexes the digit. Pelvic limb muscles, the main ones, we have the gluteal muscles, which extend the muscles of the hip. We have the hamstring group back here. They extend the hip joint, and they're the main flexors of the stifle joint. They include the biceps femoris, which is on the lateral surface of the rear limb, the semimembranosus, and the semitendinosus. These are back in the back. These are the two major muscle groups, the semimembranosus and semitendinosus, that you want to push together because when you give an injection, because in between them runs the sciatic nerve. There's also the quadriceps group. It's a four-headed group of muscles that goes over the femur, quadriceps femoris, and it's the main extensor muscle of the stifle joint. We have the gastrocnemius muscle that is the extensor muscle of the hawk. And the gastrocnemius muscle, the tendon of that is called the Achilles tendon in humans. So there's some myth behind that word, and you can actually feel your Achilles tendon just above your ankle on the back of your foot, and that comes up into your calf muscle. That's your gastrocnemius muscle. Muscles of respiration. The function of the muscles of respiration are obviously to increase and decrease the size of the thoracic cavity. Now, if you'll remember or learn this in just a little bit, the thoracic cavity is under negative pressure. So if we increase the size of it, it allows the lungs to fill. There are inspiratory muscles to fill the lungs and expiratory muscles to squeeze the air out of the lungs. The inspiratory muscles are the diaphragm, which is an internal muscle, and then your external intercostal muscles. Inspiratory muscles, diaphragm, and external intercostal. Expiratory muscles are your internal intercostal muscles and your abdominal muscles. The intercostal muscles are those muscles that are in between the ribs. Skeletal muscle cells are very large. In fact, they're very long. They're multinucleate because they are so long. They need lots of nuclei within them to create energy for the cell. They have there are numerous myofibrils within this cell that are composed of actin and myosin. They're tiny little fibrils, tiny little fibers in the cell with actin and myosin. And it forms a network within that cell of called a sarcoplasmic reticulum. And these have T-tubules that allow communication of minerals and different chemicals to go in between the sarcoplasmic reticulum. So if you're looking at a large muscle, here's the tendon, here's the muscle. There are different sections. The epimysium is the outer connective tissue sheath around the whole muscle. We have the perimysium, which is in between each of the fascicles. The fascicles are made up of muscle fibers or muscle cells. Muscle fibers are muscle cells. We also have in between these individual muscle cells or muscle fibers is the endomysium. So this muscle fiber, so you take this out and you look at it very closely, and within that muscle fiber or muscle cell, you see multiple nuclei. And inside of that muscle fiber, you have a myofibril, so a very, very small myofibril. And on that myofibril, you'll see dark bands and light bands. There's the A band and the I band. And within those myofibrils, we have teeny tiny myofilaments. These myofilaments are what make up a sarcomere. They are composed of myosin, myofilament, and actin. And just the way they're lined up will create different zones within the skeletal muscle, which make it look striated or striped. And these different zones have different activities. And we can explain this more. There's a video that really explains it very well. And basically what is happening is that these two lines under certain conditions will pull together. And as each of these lines pull together, each of these lines pull together, each of these lines pull together, it will contract the muscle. So it happens at a very microscopic level, but it affects the whole muscle when it is done in conjunction with each other. So within that skeletal muscle cell, we have A bands. These A bands are thick myosin filaments. The I bands are thin actin filaments. There's a dark line in the center of the I band, which is called the Z line. So here's the Z line in the center of the I band. The I band are those thin actin filaments. There's a disc that is the attachment site for those actin filaments. Now, if you are trying to pull on something, say you have your dog on a leash and you are pulling your dog, you have to have that dog provide some resistance. If that dog provides some resistance, that's what gives you the pull. If the dog just comes along with you, there is no pull, no resistance, and you don't get a contraction, if that makes sense. So you have to have some resistance to get that contraction of the muscle, and that resistance is that Z line where, or the disc where you have the attachment site for the actin filaments. A sarcomere is what we call the basic contracting unit of the skeletal muscle at the very, very smallest level. It is considered the area from one Z line to the next Z line. Each myofibril is made up of many sarcomeres, and they're all lined up end to end. When those, and I'll say this again, when you have those discs, Z line to Z line, and in between those Z lines, that's contracting, you'll get a shortening of that A band. And that is what gives you the, if you do it all together, the overall contraction of the muscle. So there is, in order for this to happen, there's a neuromuscular junction. Our skeletal, all of our muscles are innervated. And so it's where the nerves and the muscles come together. There is a place called a synaptic space. And within that space, that's where synaptic vesicles, which are sacs at the end of the nerve fiber, they contain a neurotransmitter called acetylcholine. Acetylcholine will be transferred to the receptors on the muscle, and that will cause a contraction. There's also acetylcholinesterase. Esterase is something that erases the neurotransmitter from the receptor, and that acetylcholinesterase enzyme in the synaptic space removes acetylcholine and puts it back into the nerve so it can fire again. A motor unit is when you have one nerve fiber and all the muscle fibers that it will innervate. That's called a motor unit. Muscles that make small, delicate movements have only a few muscle fibers per nerve in each motor unit. So your little fingers make small, delicate movements. That's because each nerve fiber just has the capability of doing just a few muscle fibers. With large, powerful muscles, they may have 100 or more muscle fibers per motor unit. So your large thigh muscles, when they're moving, they've got nerves that are in charge of a lot more muscle fibers. All right, so we talked a little bit about the connective tissue layers that surround these muscle fibers. Let's go over those again. They hold these components of the muscle together, and they contain blood vessels and nerve fibers that supply the muscle fibers. They are continuous with the tendons or aponeuroses that connect the muscles to the bones to other muscles. So everything is connected within the body. And these connective tissue fibers or layers surround the whole muscle as a whole, which is the epimysium. But it starts with the endomysium, which is composed of fine reticular fibers, and it surrounds each little muscle fiber, each myofiber, each muscle cell. Then those groups of muscle fibers together are held within a fascicle. Okay, so this little tiny reticular fibers in this endomysium around each of the muscle fiber, and then together we have a fascicle. And a paramysium is composed of reticular fibers and thick collagen fibers, and that surrounds the fascicle. Then the epimysium is a fibrous layer composed of tough collagen fibers, and it surrounds groups of muscle fascicles, and that's what makes up the skeletal muscle. The initiation of muscle contraction and relaxation. So that happens when a nerve impulse reaches the end bulb of the motor nerve fiber, and the acetylcholine is released into the synaptic space. The acetylcholine molecules bind to receptors on the surface of that sarcolemma, okay, that individual unit, and the impulse travels along the sarcolemma and through the T-tubules into the interior of the cell. Here's your T-tubule on either side. So contraction and relaxation in a smooth muscle cell, we have this picture of the cell and what's happening. Do I need you to recognize every step of the process? No, I don't. What I need you to recognize is that there is a stimulant, stimulating ligand, something that connects with the receptor, and it causes things to happen. And what it causes to happen is calcium comes into the cell. We need calcium in the cell to inactivate the contraction, okay? And this contraction does take energy. So you knew this. You knew that it takes energy to contract your muscles. This is nothing new for you to remember. It does require calcium to be available for muscles to contract, And that's all muscles, skeletal muscles, smooth muscles, cardiac muscle, and it's got to be the right amount of calcium. When we have calcium come into the cell, at some point we need the cell to work to remove the calcium from the cell so we can have this process start again. And so that's what takes up energy. So the impulse reaches that sarcoplasmic reticulum within the cell. Calcium ions are released into the sarcoplasm, which is the cytoplasm, and calcium diffuses into the myofibrils and starts the contraction process. Then the sarcoplasmic reticulum pumps the calcium out again, and calcium is pulled out of the myofibrils. Contraction will stop, and the muscle will return to its original length. So a muscle will twitch, contract, and relax. That's the normal way that we will get a muscle contraction. The mechanics are that we have a relaxed muscle fiber that has actin and myosin filaments that they slightly overlap, but they don't come into a lot of contact with each other. But when they're stimulated to contract, we have cross bridges on the myosin filaments that slide back and forth. So this is what makes it kind of look like a factory happening, where you have those myosin filaments pulling on the actin filaments and bringing them toward a center point so that contracts from Z disc to Z disc. Actin filaments on both sides pull toward the center of the myosin filaments, which shortens the sarcomere, and the shortening end to end of all the sarcomeres in a muscle results in that muscle contraction. It's an all or nothing principle. When it's stimulated, an individual muscle fiber, one cell, either contracts completely or not at all. The nervous system controls the number of muscle fibers it stimulates for a particular movement. So if you have small, fine movements that are required, then you only have a few muscle fibers that are contracting, but they're all contracting or they're not contracting. Larger, more powerful movements require contraction of many muscle fibers. So there are 3 phases of muscle contraction, and this becomes, it's interesting, but it also becomes important when we think about certain diseases. There's the latent phase, which is the time between the nerve stimulus and the beginning of a contraction. So we've got the nerve stimulus, and we're waiting for those myosin and actin to completely contract or to start to contract. And that takes about 10 milliseconds. The contracting phase lasts about 40 milliseconds. So it takes 40 milliseconds to pull the actin filaments together. And then the relaxation phase takes about 50 milliseconds. So the latent phase, from the time you get the impulse to the time you're in full contraction, that's 50 milliseconds. And the relaxation phase is about 50 milliseconds as well. So from the time you get the impulse to the time that you're completely relaxed and can accept another impulse is 100 milliseconds. So the maximum, like I said, the maximum contraction efficiency, another way to put that, occurs if the nerve impulses arrive about 0.1 second apart, because you have to have this time to allow it to go through the latent and contraction and all the way to the relaxation phase. If you get another impulse before that 0.1 second, it's not going to be able to contract again. So this results in a series of complete muscle fiber twitches. So twitch, 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 twitch. Careful timing of this allows that muscle to contract smoothly. We don't feel those twitches because they're happening so fast. But if we don't have careful timing, we will have movements that are a little bit choppy and a little uncoordinated. So it is important that acetylcholine is going through at a certain time period, that we have acetylcholine esterase that's breaking it apart, allowing it to come back to the nerve. And all of that has to work in a very smooth manner, and it's got to be 0.1 second apart. And if it's not, then we have problems. Remember, ATP is providing energy to allow the sliding of the actin and the myosin filaments. We also rely on creatine phosphate to convert ADP back to ATP so we can use it for energy again. Glucose and oxygen help produce ATP and creatine phosphate. So creatine phosphate is something we look for in blood tests, and so it's important to remember that. Glucose is stored in the muscle as glycogen, and then we can use it as stored energy. Oxygen is stored as myoglobin. Anaerobic metabolism is going to be used if the oxygen need exceeds the oxygen supply. So if you are running, and you have run out of oxygen, you've run out of energy, then we have lactic acid formation because we have anaerobic metabolism happening. Lactic acid accumulation causes discomfort. This is why you are sore after working out really, really hard, because you've gone into anaerobic metabolism, which produces lactic acid, which burns your muscles. So if you were ever wondering what happens there, this is what happens. And in some cases, it's actually not a bad thing. It helps you to build new muscle, and that's why you want to feel the burn, right? But at some level, it can be damaging, so you can overdo it. Heat production. Muscle activity generates heat. Panting or sweating are mechanisms to eliminate excess heat. Shivering is a reflex that causes spasmodic muscle contractions that increase heat production. So our body knows how to create heat as well. All right, so we talked about skeletal muscle. Cardiac muscle and smooth muscle contract in similar ways, but because they are arranged differently, it allows the arrangement of those muscles to contract in a slightly different way. So the cardiac muscle has small cells, and they have a single nucleus because they are smaller cells. They have in between them intercalated discs, and this is the attachment between the cardiac muscle cells, and that's like the Z line or the discs where the actin filaments are attached and cause that contraction. These intercalated discs allow transmission of impulses from cell to cell so that we have a coordinated contraction for large groups of cells. That went too quickly. The physiology of the cardiac muscle. Cardiac cells contract without any external stimulation. There are groups of cardiac muscle cells that contract at a rate of the most rapid cell in the group. So there's one cell that leads the charge. Contractions are rapid and wavelength. And so you need to think of this in terms of we need this heart to work in a certain direction in a certain way. So it's pushing blood in front of it. The contraction needs to happen to push blood in front of it and out of the muscle. So the physiology of the cardiac muscle, again, we have a cardiac conduction system. Starts with the sinoatrial node, the SA node, which generates the impulse that starts each heartbeat. And this is located in the wall of the right atrium, and we'll cover this again when we talk about cardiac anatomy. That impulse follows along a controlled path through the conduction system of the heart, and the structures in the system transmit, delay, redirect this conduction in order to get the heart muscles to beat in the right way. So nerve supply to the heart, the heart does have nerve supply, so it starts with an SA node just located within the heart, but it also is innervated by nerves by both the sympathetic and parasympathetic nervous system, and it helps to kind of regulate the heartbeat a little bit. Sympathetic fibers stimulate the heart to beat harder and faster because it's part of that fight or flight response, whereas parasympathetic fibers inhibit cardiac function and cause the heart to beat more slowly and with less force, so rest and digest. In smooth muscle, grosso modo, there are two main forms. There's visceral smooth muscle, which are large sheet of spindle-like cells in walls of some of the hollow organs. They contract and relax. The multi-unit smooth muscle are small, discrete groups of cells. So these are small, spindle-shaped cells with a single nucleus. The actin and myosin filaments are arranged as small contractile units that crisscross the cell. So when they contract, they squeeze the cell smaller. They're attached at both ends. Remember, we have to have some type of attachment to cause that contraction, and that's at dense bodies. And they correspond with the Z lines of the skeletal muscle or intercalated discs of the cardiac muscle. So see how it changes the shape of the cell to make it smaller. Visceral muscle found in the walls of many internal organs, stomach, intestines, uterus, and urinary bladder, and it contracts in large rhythmic waves. It contracts without external stimulation because it reacts to stretching by contracting more strongly. So if it stretches, it's going to contract even more strongly because as things get bigger, we need more of an effort to remove things from that organ. It's innervated by nerves from both the sympathetic and parasympathetic nervous systems. Sympathetic stimulation in general decreases activity, while parasympathetic stimulation will increase activity. Organs are mainly used for rest and digest, or these visceral organs that we're talking about, mainly used for rest and digest versus fight or flight. Multi-unit smooth muscle are individual smooth muscle cells, like one cell or a small group of cells that are found in small, delicate contractions. That's where small, delicate contractions are needed in the iris of the eye, where your pupil will constrict or dilate, and the walls of small blood vessels. And their contraction actually requires autonomic nervous system impulse, so sympathetic or parasympathetic nerve impulse. All right, so I have a couple of videos. I'm going to post them. These are great and fun ways to learn a little bit more about the muscles. It may explain a little bit better than I have. If you have any questions, please bring them to class. Thanks for watching.